Discovering Vibrio pectenicida, the culprit behind sea star wasting disease. Sea star wasting disease has long mystified researchers, but new revelations point directly to a bacterial culprit, Vibrio pectenicida strain FHCF3. This discovery could fundamentally change how we approach marine ecosystem health and sea star conservation. But here's where it gets controversial. While prior studies linked viruses or environmental factors to the disease, pinpointing this particular Vibrio strain shifts the spotlight toward bacterial pathogens, raising questions about the complexity of marine disease ecology. Researchers have made their comprehensive datasets publicly accessible to facilitate further investigation. Metatranscriptomic and 16S rRNA gene sequencing data are stored in the NCBI Short Read Archive under bioproject number PRJNA 115080. Additionally, the complete genome sequence of Vibrio pectenicida strain FHCF3 is deposited in the NCBI GenBank repository, accessible through accession number JBL-ZMR-00000, with accompanying raw sequencing reads also archived under bioproject number PRJNA 1232168. The full 16S rRNA gene sequences pertinent to this strain are also available in GenBank, accession numbers PQ700178 and PQ763222 through PQ763229. Supporting source data for this study can be found attached to the original publication. The computational code developed and used throughout this research has been archived on Dryad, ensuring transparency and reproducibility for future studies exploring marine pathogen genomics. A rich body of prior research contextualizes this groundbreaking finding. For example, Eisenlord et al. 2016. Analyze the effects of population size and temperature on sea star mortality during a major wasting disease outbreak. Hewson and colleagues, 2014, uncovered a denzovirus associated with sea star disease. Yet this new finding invites us to reconsider the pathogen's role versus bacterial infection. Dawson et al. 2023 provided a decade-long perspective on sea star diversity and disease dynamics, underscoring the complexity of the issue. Other pivotal studies illustrate the widespread ecological collapse caused by such diseases. Harvell et al. 2019 identified large-scale predator decline. This research doesn't stand alone. It builds upon decades of marine microbiology, viral ecology, and bacteriology studies, illustrating the intricate interplay between microbial communities, host species, and their environments. One key takeaway is that microbial dysbiosis, disturbances in the natural microbial balance, often precedes visible disease symptoms, hinting at potential early intervention points. The methodologies embraced integrated cutting-edge genomic sequencing. Bioinformatics tools such as Diamond for protein alignment, KIME2 for microbiome analysis, and advanced phylogenetic software to parse evolutionary relationships. These technical tools have not only accelerated data analysis, but also improved accuracy in identifying pathogens within complex environmental samples. Acknowledgements are due to a wide network of collaborators and institutions that facilitated the fieldwork, laboratory analyses, data curation, and funding. From permit acquisition to expert advice and logistical support, the multi-institutional effort reflects the scale and urgency of addressing marine disease impacts. Contributing authors represent diverse expertise across ecology, microbiology, marine biology, and computational sciences, hailing from prestigious institutions in Canada and the United States. Their combined efforts exemplify collaborative science aimed at unraveling marine pathogen mysteries. The authors emphatically declare no competing interests, reinforcing the credibility and objectivity of the research findings. Given this evidence, a critical question arises. Does focusing on Vibrio pectenicida as the primary pathogen clarify the root causes of sea star wasting disease, or might it overshadow other contributing viral or environmental factors? Could the disease ultimately be a multifactorial syndrome requiring holistic management strategies? We invite readers to share their perspectives. Is it time to rethink marine disease paradigms, or does this reinforce established knowledge? Your thoughts could shape the next wave of marine conservation debates. Solved. The mystery killer behind 5 billion starfish deaths, marine bacteria discovery. A decade-long enigma that left billions of starfish along the Pacific coast of North America in a state of mass mortality has finally been cracked by scientists who have identified the lethal agent behind the devastation. In 2013, a mysterious killer swept through sea star populations, decimating an estimated 5 billion of these creatures from Mexico to Alaska, with over 20 species still reeling from the effects. Alyssa Gaiman, a marine disease ecologist at the Hakai Institute in British Columbia, Canada, who played a pivotal role in pinpointing the cause, described the scene as truly gruesome. Healthy sea stars, also known as starfish, typically have puffy arms that extend straight out, but the wasting disease that emerged over a decade ago caused them to develop lesions, followed by the shocking detachment of their limbs. The sunflower sea star species was hit hardest, with a staggering 90% of its population wiped out in the first 5 years of the outbreak. After numerous dead ends and misleading clues, scientists are now confident that the culprit was a bacterium that also infected shellfish. This revelation, published in the prestigious journal Nature, Ecology, and Evolution, answers a long-standing question about a very serious disease in the ocean, according to Rebecca Vega-Thurber, a marine microbiologist at the University of California, Santa Barbara, who was not involved in the study. Previous investigations had missed the mark by focusing on the wrong type of virus or by examining samples of deceased sea stars that no longer contained the bodily fluid surrounding their organs. However, the latest study involved a meticulous analysis of this fluid, known as coelomatic fluid, and identified the pathogenic bacteria Vibrio pectenicida. The demise of sea stars had a domino effect on coastal ecosystems. With predators like the sunflower sea star out of the picture, sea urchin populations surged. In Northern California, this led to the destruction of approximately 95% of kelp forests over the past decade. Critical underwater habitats, often referred to as the rainforests of the ocean. Callum Duncan, head of policy and advocacy at the Marine Conservation Society in the UK, emphasized that the overgrazing of kelp by sea urchins highlights the delicate balance of the ecosystem. He added, "The ocean is our greatest ally in combating the climate crisis, and we must do everything in our power to enhance marine ecosystem health." Scientists are optimistic that their discovery could aid in efforts to save sea stars. Research is underway to determine whether relocating the animals or breeding them in captivity for later reintroduction to areas with depleted populations is feasible. They are also exploring whether treatments like probiotics could bolster immunity to the disease. Governments are currently convening in Geneva to negotiate a global plastics treaty aimed at preventing plastics from disrupting marine life, fishing, and tourism. The UK government has called for a bold and ambitious global agreement to end plastic pollution by 2040. Marine Minister Emma Hardy stated, "The UK is taking a leading role in advocating for an effective treaty that ensures the sustainable consumption and production of plastics, addresses problematic plastic products, and paves the way for a circular economy."